Hi, sir. This is Govind, and I'll discuss about a paper on bacteria of lambda and how it evolves when its host genes are tweaked. So, as we know, viruses are not metabolically very active themselves, so they use hosts machinery for replication of their genome and increase their number. So, in this study, the authors wanted to test the hypothesis that how viruses co-evolve with the host. So, if we if we tweak the genes of host, which were very important in viral replication, so how will the virus evolve in such a fashion to survive? So, for this experiment, they choose a very simplest model system to test this hypothesis, E. coli and its bacteriophage lambda. So, lambda uses E. coli enzymes DNA J and DNA K for its replication, along with its own proteins O and P. So, in this picture, you can see lambda protein O binds at the ori site, then recruits the protein P. Then P binds the host's helicase DNAB. Um, After that, E. coli proteins DNA J and DNA K join the complex as shown in the picture. As you can see, DNA-K is attached to phage P protein, so it helps in hydrolysis of ATP. So, so that the replication complex could start. Actually, the ATP hydrolysis make the conformational change in P that initiates the replication. So, if you see the DNA J doesn't play any role directly, but can help in recruitment of DNA K. So, to test this hypothesis, they knocked out the DNA J and DNA K genes one by one and observe what viral genotypes and phenotypes changes takes place. So, in this case, a knockout of DNA J and K doesn't impact the E. coli growth very much. So it is very good thing. Uh, Also, by common sense, we can conclude that as DNA J and DNA K are in contact with uh, the lambda O and lambda P proteins of the phage. So the knockout of the DNA J or DNA K will change will it will generate mutations in O and P proteins if anyhow the lambda phage evolves to adapt the new genetic system of the modified E. coli. But it's not the case. Why it's not the case? We'll see further. So now how we'll test our hypothesis. So we have to do some experiments. So these are the some experiments we'll do. So evolution experiment, competitive fitness assay, growth rate assay, mutualism assay, mutation discovery, mutation reconstructions. So we'll see them all one by one. So first we'll see the evolution experiment. So the authors said 12 population of lytic strain of lambda, in which they put six of them in wild type E. coli as a controls, and the rest six of them in DNA-J knockout E. coli. So to check whether the lambda is replicating in E. coli or not, they isolated fag at the end of each cycle, separated the debris, and again reinfected uh, the E. coli and repeated this process several times. And then they did the fast density measurements at the end of first, fifth, 10th, and 20th cycles. So actually, they counted the plaques to ensure that the phages are still alive in the bacterial cultures. Also, to confirm the lambda abundance, they did a PCR test for the specific lambda genes. So, as predicted, all the wild type E. coli let the lambda replicate and were susceptible to lysis even after many cycles. So on the other hand, DNA J knockout E. coli didn't let any phage replicate. So only phages that would have evolved, only they can survive in DNA knockout environment. So the authors found two populations that could survive in DNA environment, DNA knockout environment by adaptation. So they were named DNA-J1 and DNA-J2. So as you can see in the graph, only two strains of lambda managed to survive in DNA-J knockout environment of E. coli. So they are behaving like they were grown in wild type E. coli. But actually these two were found in DNA-J knockout E. coli. So the next test we are going to do is a competitive fitness test. So once we separated the evolved phages, now we need to check whether they are fit enough compared to their ancestral wildlife phages. So to check this, we injected the LAC-G gene in ancestral lambda phage CI26. So which gives blue color plaques as we are using DH5 alpha strain of E. coli. So only source of LAC-G is from modified phage strain. So marked ancestors give blue plaques, whereas evolved genes gives clear plaques. So by calculating the plaques number for each one after rating, we can calculate the relative fitness. So here in this picture, you can see the evolved strains have higher fitness than the wild type ones. So yeah, for the both DNA-J1 and DNA-J2. So similarly, in DNA-J knockout E. coli, also the survival rate for the DNA-J1 and DNA-J2 are completely significantly very higher than the wild type ones. So now the next text we will do will be the growth rate assets. So growth rate measures changes in population size over time. So in short, it's a measurement of combined effects of birth and death. So we test growth rate either in liquid culture uh, or by a more common assay, that's plaque assay. So where phages are plated on loss of the host cells. So actually, here you can see the growth rate is positive only after a certain amount of phages. So you need a minimum threshold amount at least in order to infect. So the next test experiment we are going to do is the mutualism test. So this assay is done to check whether the evolved lambda um, has enhanced ability to cough rate or not. So if it is so, then the marked wild type phages should survive better in the presence of DNA J1 as compared with when grown in presence of another wild type phage which isn't marked with like G1. So in this image, you can see the marked phage is surviving better in the presence of DNA J1 or any DNA J2 because DNA J1 phage increases the lysis timing in DNA J knockout E. coli, gives phage more time, even the wild type phage more time to a spontaneous DNA K hydrolysis and DNA replication. So also the increased number of proteins of O and P concentration. So increase the chances of successful replication. So here you can see the when the wild type were grown with DNA J2, its survival rate is much higher than when it was grown with uh, not non-marked ancestral strain of lambda. So the next thing experiments we are going to do is like once we discovered all the evolved strains, tested its fitness and mutualistic behaviors. So now obviously we will want to know what genetic mutations led to these changes, and can recreating these changes in a wild type lambda phage uh, again recreate those behaviors or not. So to, in these curiosity, they sequence the lambda O and P genes from DNA J1 and DNA J2 because they had predicted uh, they must be on these genes as they were in direct contact with DNA J and DNA K. But they didn't find any mutations in these genes. So then they did the whole genome sequencing, and surprisingly, they found mutations at three other locations, J, S, and ORF314. So they recreated single mutations for each of the three wild type, each of the three in wild type, and observed the growth rate. So they also created double mutants for J and S. So in all the cases, the growth rate for the modified ones are higher than the original wild type one. So, so as J codes for the tail fiber protein, so mutations in this may affect adsorption. So, to test this hypothesis, they compared the adsorption rate of CI857 with the adsorption rate of modified lambda having only J mutations. So also to test the function of S mutation, they compared the modified lambda phage having only S mutation with the wild type one, and then noted the lysis timing. But because these replicates so slow, so to fast forward the process, they give heat shock to the lysogens. So the C1 get denatured and the lysis takes place faster. So we can compare the wild type and the modified ones lysis rate. So at the end, they got the results. So in this adaptation process, phages incorporate three mutations, J, S, and ORF314, among which only J and S mutations were useful. So J is actually a host recognition protein. It codes for tail fiber. So its increased adoption rate. So its infectivity is high even in low concentration compared to wild type. Um, uh, because the more the adsorption on the surface of bacteria, more highly effective it will be. So mutations in S increases the lysis timing, so which gives phage P protein more time to spontaneously hydrolyzed by DNA of the E. coli in DNA J mutants. So at the end, so these mutants were not in genes of for proteins that directly interact with the missing network element, but were in the genes impact the broader features of the virus life cycle and production. So we can say the host and viral relations is not that simple. So we have to think broader. Uh, and